All right. Okay. We're gonna start. So thanks everybody. Thanks everybody for coming. As you know, so this is we will open now the 2019-2020 academic year for the Quantitative Systems Biology Center. And so this year we're going to have a novelty, a new thing, and that is this series of conferences of seminars is going to be linked to a class, so to a CPB class. You know what is CPB? Chemical and Physical Biology? Okay. And we have a number of CPB students here. Can you raise your hand? So, as you know, CPB is a program that collects a number of, should I say, quantitative biology students, and he has PhD tracks in imaging, in structural biology, chemical biology, and in systems biology. Okay, and so together with Bruce Damon, and after the suggestion of two students from CPB, believe it or not, so Tavia Sherman and Cody Heisler. So neither myself nor Bruce thought of this earlier when they came. They saw, proposed, and we did it. And so, we're gonna have. So this is gonna work this way. It's kind of like in Bill Holmes was telling me that this is the structure that's also used in physics for colloquium. Yeah, something like that. And so we're gonna have the seminar on Friday from a speaker, either from the outside, like today, or from the inside. And then so the following Wednesday, there will be a one-hour class, and the class will be run by Leonard Harris, sitting in class. And so, and this year we already have 5 students, so we actually kept it 5 because we want to see how this is going to go. Okay, we still have to pull it together. And the class will be about discussing the systems approaches that the speaker presented on the previous Friday. And as we go, we're going to adjust this a little bit. For example, we just discovered today that maybe those students should have like privileged access, to the speaker when he or she arrives, and have some kind of a meet and greet, kind of thing where there is more informal discussion, either before or after the talk. And the title of this is going to be CPB 328. It's in the books CPB 328. It's going to be called Systems Thinking in Biomedicine. Okay? So thanks again, Cody, and Talia, and the other students that are going to hear Jason. He's going to connect later, but anyway. Okay, so today it is our pleasure to have Ashley Versa to open our lecture series for a couple of reasons. First of all, because she also was picked by students, not here, but you know this. Like somebody saw you present at a systems biology meeting. It's called SAC, something like that, SAC at Woods Hole last year, and so they immediately told me you would have to have Ashley come in here. And I took the class. And then the second reason is because, yes, you will see, she's an engineer by training, and she's kind of amazing. So she covers, a broad swath, of different applications in biology. And I think that would be inspirational to see, these kind of approaches, can be applied to different, I would say, very different discipline. We discussed about that last night. Okay, so anyway, Ashley, she holds 3 degrees in chemical engineering. Apparently 1 was not enough. So she has a BS from University of Oklahoma, and then a master's and PhD from the University of Illinois at Urbana-Champaign, who has been kind of a hub for systems biologists. If you look up, the CVs of other people. So during graduate school at in Illinois, she had some prestigious award from the Department of Energy. She received the Computational Science Graduate Fellowship, and then she also received a Graduate Research Fellowship from the NSF. And some of you also had those degrees, like Sarah, Gina, Kristen, and you know how prestigious those are. She also got another prize from DOE, so the Frederick House Scholar in Computational Science, for outstanding leadership, character, and technical achievement. So then she went to MIT and she was a postdoctoral fellow in the Bratz laboratory in the Department of Chemical Engineering. And now she's an assistant professor at Oklahoma State University, your native state, right? Mm-hmm. School of Chemical Engineering. And so she's a member of several different, besides being in chemical engineering, she's also hooked up with several School of Medicine kind of thing, such as the Diabetes Center, the Harold Hamm Diabetes Center, and the Stevenson Cancer Center at the University of Oklahoma Health Science Center. There's other things. There's an interdisciplinary toxicology program. She's also part of the Oklahoma Center for Respiratory Infectious Diseases. And this year she's the chair-elect for the American Society of Engineering for Engineering Educational Chemical Engineering Division. And also, I know also, Bennett tells me that she's very active at the IKE, which is a chemical engineering annual meeting where you run the systems biology session. Okay, I think there's other stuff here, but just to encapsulate everything, Ashley's also very active in educational tasks, K-12 through all the way through, of course, graduate students. And so it's my pleasure to have Ashley here with us today, and she will talk about system biomedicine and pharmaceutics, multiscale modeling of tissues, treatments, and epidemiology. Ashley.
Thank you, Vito. Thank you for the introduction. I have really enjoyed my visit here, and particularly meeting faculty from Quantitative Systems Biology Center, but also from chemical engineering and biomedical engineering. I didn't realize until I started looking through the faculty how many different application areas that are going on here overlap with my own personal research interests. And so there may be some future collaborative things that come out of this visit, but certainly a lot of very collegial interactions that came out of this and a lot of nice networking. So it's been very much a pleasure to be here, and I enjoyed meeting with students earlier today. So I look forward to hearing your questions at the end, and it's my joy to be here. So with the mention that it's a class, I'm really glad that Vito has titled it Systems Biomedicine. My work is a little bit broader than kind of what most people in systems biology look at, so I don't just focus at single cells. I look a lot bigger than that. So I am in the School of Chemical Engineering at Oklahoma State University, and my full name, Ashley Ford Versipt. I do go by both parts. It's confusing, I know. But space and not a hyphen in my last name. If you're interested in following us in the future, I am very active on Twitter. You can always reach out to me there as well. So this is my team that snapshot from the spring. I have a lot of undergraduate researchers in my lab, and at that point there were 2 graduate students. Now I have 3 brand new graduate students, and Minu is the first PhD student from my lab to graduate and leave. This work would not be possible without first startup funds from Oklahoma State University, seed grants from Harold Hamm Diabetes Center and Oklahoma Center for Respiratory and Infectious Diseases. I have a state grant that is 2/3 of the way finished. The Oklahoma Center for Advancement of Science and Technology. My undergrads who have been supported by some very generous research scholarships. I started NSF Career Award in June, and then on September 1st, I start my first NIH grant. So I'm very excited about the work that that's going to enable in my lab. I also wouldn't have been able to do this work without previous cohorts of students from my lab, and so I'll be presenting some of their work today. And then I also have 2 other very key collaborators whose work I'm going to present for you today. One is James Krall, who is an ecologist and biologist at Harvard, who did the experimental work and some of the data analysis and visualization for the bee project I'm going to talk about. And then Biswadep Day was also a collaborator on the bee project, but he is an aerospace engineer who focuses on flight mechanics of different animals, collective behavior types of things. So a very different area of systems biology. So when I think of systems, actually Leonard was talking about this before the talk, I do think about factories or manufacturing systems where multiple things are interacting together, or the classic digestive system, circulatory system, where multiple organs are playing a role together. But systems can happen at multiple scales. So you could have genes that give information together that then lead to molecular networks inside of cells or inside of small regions of cells. You can have cells of different types that interact together and form networks. Those collectively give rise to tissue function, and the tissues give rise to organs, and then those organs interact in whole systems or an entire individual. And then you can have even larger scales where you have populations of people or whole societies, and the kind of epidemiology types of approaches. We have, in my lab, focused mostly at the tissue and organ scale, so there's quite a lot of people in systems biology working at molecular types of interactions, and some of our biophysics colleagues and people doing molecular dynamics and other sorts of atomistic and DFT types of approaches. And there's a lot of very elegant work going on in the intracellular networking level. I was very interested in tissue level types of interactions and how can we take information about biopsies or clinical types of measurements on blood pressure or blood concentrations or urine samples, how can we map that to information happening at this level? And so I was very interested in bridging these scales with different types of quantitative techniques. So today I'm going to share with you a few vignettes. I'm not going to do a deep dive in these stories. I'm not going to inundate you with all the equations or all of the codes on these things, but I wanted to give you a flavor of different types of topics that we've approached in my lab, and these are all work that we've published in the last 5 years with my team and collaborators. So one area is immunology and immunotherapy, and I'm just going to tell you a short story about one of our published works on tuberculosis, but we have some ongoing work with arthritic inflammation and with the lung and delivering aerosols there and thinking about stimulating the immune system and dendritic cells. So I won't talk about any of that work. It's very early, but those are other things I'm interested in. We also have worked on cancer metastasis with several different students, but one in particular was very productive as a master's student, got 2 papers published, and so developed a model for the onset of metastasis and some of the key chemical and physical processes there. And I'll tell you about that. Through a mathematics center, there was a invitation for a collaborative meeting where people, experimentalists had data sets and computational folks came together with tools, and they formed partnerships and worked on collaborative projects. And so that's how my bee pesticide project came about. And so very open to collaborating with people. There's another project related to fertilizer effects and controlled release that has been published while I've been faculty, but I won't tell that story today. But I will tell you about this agent-based modeling approach we developed for bees. And then the biggest area of my lab has focused on the effects of diabetes on the kidney, and specifically different cell types that are impacted biochemically, but then also biomechanically, and how those cells interact. That's led to a number of different technical papers, but also some educational initiatives, and I'll briefly show you some of the outreach work that we do, leveraging some of those software tools that we develop. And then we've also done some stuff with just blood flow regulation and kidney physiology. So here's the graphical outline for the talk, and I'll spend just a few minutes on each of the first 2 and then dive in more deeply on the latter part. So the things that are the relevant pieces to know for our first immunology project was related to tuberculosis. And in tuberculosis, you have a bacterium, which is represented by these red cylinders here. And bacteria comes in, and a patient, typically their immune system responds to that bacteria and can either immediately have active tuberculosis disease or can barricade the tuberculosis bacteria and form what's called granulomas. And so the immune system with many cells responds and forms basically a fibrous fence around these clusters of cells, and that spherical cluster is called a granuloma. And so there are many people who've studied the infection of an individual with TB and whether they would develop active TB disease or latent TB disease. There's a lot of work, particularly from the University of Michigan, on the modeling and experimental side on that. And so what is known, though, is that these granulomas, once successfully established, the person doesn't have active TB, it's still lingering. It's not dead. It's more dormant for long periods of time. And patients can, if later they have some immune compromising event, can get active TB disease many years after their initial exposure. And so these cells are just quiescent, and they can transition to active TB if this extracellular matrix or fibrous collagen network around the latent TB granuloma is breached and allows the bacteria to escape. And so we were very interested in what processes would perturb a latent TB state to get active TB. And clinically, it's known like things like aging, diabetes, HIV, or other types of infections could lead to this transition. We were interested in the dynamics and the magnitude of those perturbations. And so we wanted to have a model that could allow for this transition after long, steady latent TB into the situation of active TB. And so we see that the bacteria leaking is a major part of that, and something about this collagen degradation. So we looked at factors that would influence that, and that would be an upregulation of MMPs. And so these tuberculosis bacteria can actually signal and get MMP to increase, that basically allows them to penetrate and break apart this collagen network. And so we built a model that had added to an existing model of the steady state to have the bacteria leakage, but depended on how much collagen was actually there in the local environment. And we have a collagen remodeling equation that dealt with how much collagen is being recruited and then how much collagen was being degraded, enhanced by the presence of MMP. We did a lot of different things with that model, but one of the key things we explored was, what if in HIV you have the initial T cell population in the immune microenvironment has changed due to the presence of HIV infection, and so that's what this curve is representing. So this is our input to the system. Then we saw that the collagen responded by eventually degrading, and that the bacterial leakage increased dramatically, and this is a log scale, so it's definitely leaking out in that scenario. So that's one example of work that we could do in this space where there's a tie between the remodeling of some sort of extracellular tissue and an immune response.
Was there a critical threshold for T-cell mediated release?
We didn't explore that explicitly, and that's one where there was a short time frame for a seed grant and collaborators with no funding, so that one didn't go as far or as deeply as we'd like it to go. We have lots of questions, though, that we would love to answer if we had more support on that specific project. So,
Actually,
yes?
I guess what do you mean by simulated HIV?
So in HIV, we know that people have fewer T cells, and so we artificially had the T cell population go down. So this one is a simulation input, that these are then the outcome for our model.
Do you take into account like CD4, CD8?
So we did have CD4 and CD8 T cells in the population. This is the precursor, I believe, to the CD4 plus population. Yeah. The next vignette is on cancer metastasis. So the idea here is that we have a primary tumor, and tumors will, to spread, often need to remodel their environment in order to enhance the migration of cells away from the primary tumor into the blood vessels and away to other metastatic locations. And so there are various chemicals involved here that involve secretion. We represented MMPs here by little Pac-Man. They break apart the fibers, and then we have these little bow ties, the locks. They are cross-linking and reorganizing the fibers, organizing the remaining fibers into structures that are that facilitate transport of or migration of cells along those fibrous tracks. And so all these chemical interactions and the physical forces are important for the remodeling and the migration of these cells at the onset of metastasis. And so my student worked on making a model for this process. There were existing models that dealt with MMP, but not ones that involved that cross-linking and the remodeling influence of, or the realignment process. And so that was the contribution that we added to this project. So we first did a 1D continuous version that took compared, we presented it in the same way that an existing model did, but we had added this other feature of having locks in the system, and showed over time that we could have different cell populations based on the locks. We also then looked at doing a 3D or 2D, this is a 2D cross-sectional view of a tumor spheroid with different types of environments and how it would respond, or how the cell migration would respond if the cells preferentially wanted to go to fibers that had been realigned. And so that work was very recently published. So with that, we looked at effects of having a randomly distributed extracellular matrix versus having a very uniform single density or single stiffness type of extracellular matrix, and the effects of that over time on different migration patterns of cancer cells. And we saw differences in both the average displacement, how far did they move, but then also how sort of symmetrically did they move, and did some pockets of them move more quickly than others. And so you see kind of more heterogeneities in the movement here versus this was pretty radial in the movement patterns. We also, and the time is more of a simulation unit, but 10 days of migration speed at what we had. And then we also compared what if we changed the average density or concentration of our fibers, if it was a very open fiber network versus a very closed fiber network, how did that influence the migration patterns of our cell networks? And we did that for both the uniform and the randomly distributed, and we saw some different patterns. Note these are different scales, so this is a very much larger effect than this. We also explored the effects of different initial conditions. And the state of this work is that we know this is all theoretical. We like to compare it to experimental cell migration studies. And so my students have identified migration data that's available, but my undergraduate student on this project just stepped off of the project last week. So I'm looking for new students to continue this line of research. I'm very excited about it. I think there's important things going on at Vanderbilt and data that's collected by some of the labs here that can be used in ways that connect nicely to this particular project.
Is it just, there are several different types of MMPs, right?
Yeah, so we were looking at, I think MMP1 in this particular one, that the MMP that targeted that particular type of collagen. So we were looking at one type of MMP and one type of collagen, recognizing that in different tumors, those rates might be different for the different combinations or in different types of extracellular fibrous materials.
Did any of your results, like MMP inhibitors are not very successful?
So we didn't change anything about the inhibitor amounts. But we were very interested in how does cross-linking in particular. So we did some comparisons of what if we just had MMP and degradation versus if we had cross-linking, and could we get migration patterns that were consistent with, qualitatively consistent, and by having the LOX part of the cross-linking, they were much more consistent. So that was our main focus, was thinking about the cross-linking component. So shifting gears, so those two approaches, one had ordinary differential equations. The second one had a hybrid approach with ordinary differential equations or partial differential equations and throwing on a layer of stochastic agents on top of it with the cancer cells. This work is completely agent-based, and we actually used MATLAB to do this, and my collaborator came to us at this collaborative meeting with this data. So bees exposed to pesticides, and let me just, I thought I had the picture of this experimental setup. He basically has bee nests with bumblebees, and they have a video capture system on top, and he has many of these nests in parallel, so he can move his video monitoring system around or monitor them all in parallel and get a lot of data and collect that automatically through video systems. And then they have little RFID chips on the back of the bees, and so they're tagged. They can monitor where they go over long periods of time. They typically monitor them for about an hour, and so they were very interested in pesticide exposure. And this particular class of pesticides is being added to seeds and very widely used in many, many crops these days, and it's supposed to be not harmful to bees. And it's not in high doses, but it's very low chronic exposure, because the bees will then go and pollinate and continually be taking in small amounts of this pesticide. And so what my colleague showed is before a treatment that they tended to have most of their movement actively on the nest, so where their food pots were, where their brood was, taking care of their young. And then if a very low dose didn't have much effect, but even a very modest, chronically realistic dose, they had changed even just by the next day, and in this 1 hour time frame, they were much less mobile. Their activity changed, they weren't nursing as often, and they had a change in their average distance from the center of the nest. So they were staying away from the center of the nest more often. And so even though it's not killing the bees, it could have long-term impacts for the next generation if they're not taking care of their young and not doing their normal societal functions. And so he was very interested in, could we have sort of a dose-response type of model, or explore some explanation, some mechanisms for what is happening in the bees that's impacting their behavior. And so these studies would have on the order of 100 bees in them, and they do videos. Trying to find the direction of my mouse here. Not that way. Okay, it's not going to play. So if this had played, you would see the bees wiggling around, and they can take that and track and get the digital information about the bee position at each frame, and they have these small little tags on their back, and they fall off sometimes, so some of the data is you don't see. They're static because they have flipped the right side up and are stationary. Sometimes they go away because they flipped over, and the bees are allowed to leave the nest and forage and come back, but for the most part they stay inside of the nest, and you can see them. So it's a 2D orientation, even though the bees actually can go in a vertical plane. And so we model this system, so it's an agent-based system. We call it bee nest ABM. So there are other systems that look at the whole ecosystem of a bee and think much more macroscopically, but we were interested in these local sort of short-term effects, or what happens in an immediate term after this type of exposure. And so the yellow is representing the nest structure. We have the little bees. We consider we have later a force that kind of attracts them to the nest, and those a random walk sort of vector, and then we have the resultant movement based on some combination of those two. And the data we published in Science, and then we also had our first, we fit our model to the experimental data, and that's in that paper. And then we have made the software available in the Journal of Open Source Software. And so this is what the GitHub README looks like. There's, you can access the code, or you can access the paper. And then we have a lot of documentation there. We provide a very, very detailed flowchart, which I know you cannot see, but I'll tell you about the main key submodel here. And so using the empirical data, we parameterize the model with a kind of main idea. If a bee was moving and it bumped into another bee, we call that a bump, or if a bee was stationary and another bee was moving and hit it, that was a bump. And so a moving bee we called active, and a stationary bee we called inactive. And there's some probability that if you are active, you would become inactive, and if you were inactive, that you could become active. And these transitions would depend on whether you'd gotten bumped by a neighbor, or if you just suddenly got tired and sat down, or you woke up and started to move around as a bee. And so those also depended on two different types of interactions. The location, that there might be different effects. If you were already on the nest, you might be more likely to stay on the nest, or if you were off the nest, you might be more likely to keep moving. And then you might have a social interaction, that bumping, that socially moderated or modulated interaction. So spontaneous would be I just decide to stop, and bumped or socially modulated is the other mode. And so for the experimental data, we took the actual shape of what are the places in the nest and parameterized the system. And then we used those general parameters for later arbitrary defined sets of nests and probed various things. So this is just showing the analysis of probing that data for the control and the one that had been dosed with the imidacloprid, and the different parameters that we came up with. So the details of that are not critical, but my collaborator that did more of the flight mechanisms, that's his general expertise, was being able to tease these trends out of the data. And we could do things related to how before exposure, how the bees would move around, and then after exposure, we could see that they were far less mobile. These are the kind of typical types of simulation results. And the big red circle there is the queen, so we just tracked her separately. We could also artificially see the effects of changing various things in our mechanisms. So this is changing the attraction strength. How much do they want to head toward the center compared to them heading in a random walk type of orientation? So the one on the right there is obviously fake or obviously artificial, that it is saying you want to all go to the center of the nest, but it shows that importance of the strength of the interaction, that we can change that. And so we tried to change that in a way that was consistent with the data.
Are there, this is going to sound really, I don't know physiology very well. Are there mutants of bees where you could potentially turn off their social interaction and be able to decouple the parameters?
I have no idea about that. I know they were just taking bumblebees, and they probably had some very specific strain, but I don't think they were genetically engineered to ignore certain types of interactions. But they had hypothesized certain interactions, and so those were the interactions we put in the mechanism. And then this showed what if we turned on some of those different mechanisms in a simulated way, but we couldn't maybe do that in a nice genetic way. So they weren't doing a lot with they were in a neuroscience and a biology department, but they weren't actually mapping the brains of these bees and doing any sort of dissection or chemical analysis of them post-mortem, but they would observe their behaviors and use that to infer some properties. And so green means that we had the properties turned on, or that was the control, and then the red would be having both the mobility and sort of the probability pieces of how they would bump into each other or transition, and then the attraction was how much are they weighted towards the center versus doing a random walk. And so we had the right kinds of transitions if we had both pieces. But you can see here that this one captured most of it, but the other phenomena didn't. We had much more if we added both of the phenomena. So we, in general, with all these types of interactions, made an argument that they were all important. We also explored, in a separate paper, what would happen if we then explored other types of environments that weren't exactly the one that we had tested and fit the model for. What would be the predicted behavior of bees in those types of nests if they were overcrowded, or if they had different proportions of workers, or different types of resources? How would their colony size affect things like how active they were, what the difference in activity was after the dose, how active they were on their nest structure, or how diverse or how spread out they were in their movements. And so we put this out there as a tool that other ecologists could use to explore other scenarios that weren't just the nest structures that my colleague was doing. And so, the idea of giving rules to these particular autonomous agents can be applied to biology, human biology systems. It's being adapted pretty widely in systems biology for cancer cells, to give rules for how cancer cells are going to respond to different kinds of chemical signals or to different forces, or to be attracted in different ways. And so the interesting thing to me about this project was the ability to learn some techniques and apply some methods that then I could apply in new physiological contexts. And we did this in MATLAB. So we had about 100 bees, and we actually tracked each and every bee. But in a cellular system, you might represent clusters of cells, and maybe not every single individual cell. But this kind of technique could be easily applied, and has been easily applied in other application areas. So the last section. So many of us know someone with diabetes, or maybe have been impacted personally by diabetes. We know about that there's lots and lots of side effects of diabetes, but one that was very surprising to me was about how diabetes damages kidneys, and that kidney failure has two main leading causes: diabetes and high blood pressure. And diabetes and high blood pressure are often comorbidities and often influence each other. And so it's a huge medical burden. And so many people who are on dialysis are on that because of because of diabetes. And so Oklahoma, diabetes is a major, major issue. We're very much on the obesity forefront, and we have a major diabetes research center and diabetes clinical care center. And so I came from that motivation of wanting to do something that would be impactful to the health of Oklahomans, and I came upon this problem. But kind of the key thing to know is that in the kidney, there are a lot of filters for our blood called glomeruli, and that are composed of layers of different types of cells. And when these cells work properly, blood flows through vessels that then allows waste to go through the membrane, and then everything else that you want to keep stays inside. So blood cells don't penetrate this barrier, and generally proteins don't penetrate this barrier. But in diabetic conditions, you start to have several different simultaneous types of damage that will allow proteins to actually get through this barrier and into the urine. So that's a process called albuminuria or proteinuria. Glucose can stimulate these mesangial cells to grow more extracellular matrix, to get fatter and to expand and exert pressure on neighboring cells. It can allow some of the zones to be thickened. It can allow podocyte cells to deform and stretch and fall off and die in lots of different processes. But glucose is the main driver, but then there's different signaling networks that are affected extracellularly, and there's a lot of physical forces. So we've set out to try to model this damage, how you go from this healthy system by the presence of glucose, how it all works together in the onset of diabetic kidney disease. So this is showing just a view from that blood vessel outward. Normally you would have just small molecules would penetrate through this barrier, but an increasing glucose, the podocyte cells can start to pull off, and the podocytes themselves can fall off or die, and that changes the glomerular filtration barrier, where proteins start to appear in the urine, which is a clinically observable marker in the urine by urine concentration analysis. And so then they can detect non-invasively the extent of this disease, but they can't detect the onset until there is protein in the urine. So we're very interested in what's causing, how do we influence this, how do we stop this, those kind of questions. So we've been piece by piece building models for different sections of this and starting to put them together now. So we've had models that deal with the biochemical reaction networks in different of the cell types. Predominantly we focused on podocyte cells first. These are the yellow outer layer cells. They've been very widely studied in the last few years. And then we've been looking at how the transport happens from the blood vessel to the different cell types, and how glucose moves around in the zones, how proteins move around in this tissue. And then we've been taking a tissue structure agent-based modeling approach that has in the background this diffusive transport to consider the different interactions between the cells. We've focused first on the network involving angiotensin. This is a blood regulating hormone that is prevalent systemically, but it also has a very key role in the podocyte cells. And so it's primarily responsible for constricting blood vessels and raising blood pressure. And so there's some common pharmaceutical therapeutics that can inhibit the production of this hormone. And by inhibiting it, it actually, they lower blood pressure, reduce cardiovascular risk, but it's also been shown to be a treatment that slows the progression of diabetic kidney disease. So that's why we study this particular category of pharmaceuticals. And I had a couple of undergraduates in my first year who worked on building a model of pharmacokinetics for patient populations that had healthy kidney function and impaired kidney function. And that data was available, so that's why they started on that. But this does not have any sort of glucose dependency, and it only is systemic. Then we had another work that dealt with how this system is changed in podocytes, specifically in this kidney cell type. But it didn't have any pharmacokinetic effects, and it didn't have this renin-angiotensin feedback mechanism where if there's too much angiotensin, the body is supposed to self-regulate. It didn't include that effect in that model. So then we, in the most recent work that my student had for her PhD dissertation, had combined those effects in both the podocyte specific, this feedback mechanism, and the influence of this medication. And so we could do sort of classic pharmacokinetic types of things, but we could also, that would tell us how much of the hormone was there, the drug response, that kind of thing. But we could also see how that, predict how that would work in normal and impaired kidney function for these different local versus systemic systems. And we could also then say, well, what if glucose changes? Because our RAS model in the podocytes depends on glucose. And so that's our first step of having this clinical damage due to diabetes, is having this glucose. So we had two different glucose dynamics. These were data from a patient that we then just put a curve fit to, just to have what a representative patient would look like who had normal blood glucose and diabetic blood glucose levels over the course of one day. And we showed what it would look like if there was no drug dose, what happens when there's a drug dose, and then comes back to the just the cycle with the glucose dynamics and not with the drug anymore. And so when you first take the drug effects dominate, and there's not really a big difference. There is no noticeable difference down here between the normal and diabetic, but after some alleviating period of time, then the glucose dynamics start to take over. So there's periods where the drug may be the dominant effect or the glucose may be the dominant effect. So that's what we got out of that. We've also been thinking about other sections in which the transport happens. And so this is published work but from another lab, but they only considered steady state physiological conditions. And so we're very interested in what if this zone changes and changes dynamically or changes in an interactive way. What if it's not just transport? What if there's chemical interactions with the podocyte cells? And so we're scaffolding our models on top of this type of infrastructure. And yeah.
Are you looking at like adaptive dosing schedules?
No. So this stuff was not. We just said, what if we give it one dose? We weren't trying to design a drug dose. We just wanted to see the relative effects between the dose and the glucose. We could use these types of tools for dosing scheduling, but we're not. That wasn't our goal. So our goal is to build all these tools that work together. So we were able to verify that our platform works without adding the complexities of. This was verifying with a published work that we could get the same types of profiles just in color and multiple dimensions. But
so is this flow related?
So this particular problem has blood flow in the vessel that then has pressure-driven porous media transport of just the liquid through the membranes into the urinary space. So the blood is still flowing here, but glucose and plasma can make it out into the urine, and then glucose then would trigger these things, and then there'd be transport of proteins potentially as well. So the transport of proteins is highly diffusive, but the transport of small molecules depends on this hydraulic pressure from the flow. So yeah. Okay, so we've also then started doing some of this in CompuCell 3D. And so the idea there is it would propose a pixel flip. And so if we were to say that this pixel becomes part of the tissue, then there'd have to be some probability of accepting that. It's related to this energy of this transition. That energy might be associated with how much energy is between neighboring pixels of the same type, cells of the same type, or how much energy is associated with satisfying a volume constraint. Does the cell want to grow to reach a target volume, or does the cell want to migrate in a certain way? So there's a number of different things we can customize about this. And so we've made an anatomical configuration for a specific glomerular slice that has interactions with a couple of the different cell types, and we're starting to put the biochemical reaction networks into this. We did have the plasma flow part added already, and we could see kind of simple layers, but our inlet's only on this side, and then the transport of the fluid. But then we're adding in the next few weeks the chemical reaction networks that happen on top of this. And so this was showing, I don't remember that one showing, maybe that's just the velocity flow field, but this is the concentration. If the concentration's only coming in from this side, what would that look like through something toward the edge to the urinary space?
This is a really hard problem because you have multiple time scales.
Multiple time scales, multiple spatial scales.
How are you approaching a multi-time scale problem in terms of the update? Because this is an agent-based model coupled with
a PDE solver. So the agents update after something like 180 time steps on the PDE model. So the agent update is less frequent. And then
is there like a good way of choosing that number?
So there are, for the numerical stability purposes of the PDE algorithm that's used. The CompuCell 3D has a limitation that time is not the natural agent-based progression marker. It's something else that's not really time, but we mapped it to physical time based on the movement of some cells with the settings that we had. but then we know the physical time that we need for the numerical stability of the solver. So that's what was used there. We also anticipate that these would remodel the environment, but the remodeling might happen slow relative to the other processes. And so it might be okay enough to have just snapshots of those future time points rather than all of the dynamic transitions to them, or have a very long simulation that could have the chronic kind of slow transitions in the midst of more frequent modulations. So yes, the time part is challenging. All right, so where are we going with this? Recently got the career award to focus on these interactions, but having the different layers of the glomerulus with the different cell types interacting, but then also have interactions with other areas nearby, and then also to have the biomechanics. So our previous work had all been on the chemical part, but not the forces exerted on these neighboring cells. So we think about it as a virtual microscope deep down into these kidneys, because we can't do those same experiments in real time and measure chronic damage very deep within living humans. But we can use other things to validate pieces of our model. So we also, thinking about career and wanting to propose educational initiatives, we developed a graphical user interface for that pharmaceutical drug design or drug dosing for impaired and healthy kidneys. And so it was all about this angiotensin converting enzyme inhibitor and blood vessel regulation, and we talk about what is hypertension. I teach the students, these are, my target audience was incoming freshmen to the College of Engineering. We have a design program, a camp for incoming students. And I teach them that they can take raw materials, move them around, transform them often through chemical means or through separations, purifications, and get desired products. And that is really what chemical engineering is, for those of you who aren't chemical engineers. But then we had a design problem. We needed to design the best raw material. Something in the body changed the way the high blood pressure enzymes, high blood pressure regulating enzymes were transported or transformed, and the product was a broken blood pressure regulatory system. And so we needed to change the raw materials through the influence of a pharmaceutical to get back to the desired blood pressure. And so the students would play with this GUI that we made to change the dose size and frequency. So this is a dose scheduling, but this was not for research purposes. We had the research side in the background, but then we made this educational tool that students could play with, and they could have a design experience that didn't require a lot of jargon. For chemical engineering, senior level stuff, design can be quite manufacturing jargon filled, and so we focused on this kind of medicine application that most of them had taken some sort of medication in their life and could think about trade-offs with designing the scheduling of that drug. And then we had 5 different types of ACE inhibitors and 2 different kidney functions, and students could explore those. They can also try different treatment durations if they want to see what happens with one dose or one day, and they can see that it goes back up afterwards, or they can see an extended chronic treatment. And the types of output they would get would be the drug concentrations, but also if you could lower the drug. And so we told them the therapeutic target was to lower it by at least 30%. And then they have to think about some trade-offs with, do they want it as close to the line, just barely therapeutic? Do they want small fluctuations? Are they okay with big fluctuations? They have to think about some of those trade-offs. And then they can layer plots and compare different simulation scenarios. And so we have the students at the end make a presentation, and so they put some results of what they think were their best doses, and this was actual results from some student work, and so they liked, or they would present, and this would be their first presentation in college. We then said, well, this is great for incoming freshmen. I've got like 6 hours with them over 3 days, but how can we use this in other environments that doesn't require they have to be on campus, they have to be in this kind of coursework setting? Can we use this in kind of quick settings? And so there's an event hosted by Oklahoma EPSCoR called Women in Science Expo, which is a full-day event for middle school and high school girls with 1,000 students and their teachers who come to generally the Science Museum, and they do a bunch of hands-on demonstrations. They get to play with the Science Museum stuff, but then we bring our different, this is the bee simulation, and this is the pharmacokinetic simulation, and this is a different one from our lab. They can touch a computer, they can try to answer a biological question, they can think about designing something in the course of a couple of seconds or a minute or two. The bees, we just have them change that attraction strength. We say, well, is this realistic or is this realistic? How do you think the bees, do you think they would all magnetize to the center? No, probably not, but they probably have somewhere in the middle between ignoring each other or being magnetized to the center. And then for this, they can design a different drug dosing schedule. This is the using in the summer camp. And then we also have a camp called Grandparent University, which is for middle schoolers and their grandparents or someone of their grandparents' generation. And so it's a camp for alumni. They pay for it, but it's really cute, and it's a great recruitment tool for the university and great alumni relations, and so we enjoy doing it. And we put a lot more hands-on things in there. So we have some computer demos, but we also do some things with physical models for the kidney. We also do some lung things. There's these t-shirts that you can do that have, they make it look like you have, it's augmented reality stuff. They make it look like you can see your lungs and your organs, but it's really just an image on your cell phone. But we play with some of those types of demonstrations as well. We've also used these tools in kinetics. So chemical kinetics is a very natural application for this drug dosing application. Undergraduate research projects, undergraduates in my classes and in my research group have made these GUIs or have augmented them in different ways. This is the team as of last Tuesday, or a week ago Tuesday. So we just recently were awarded an NIH R35 MIRA about quantitative systems biomedicine and pharmacology related to multiscale tissue damage. And this is a very open-ended type of project to fund computational work related to the immunology multiscale tissue damage work we have. And so I do have immediate openings for postdocs, PhD students, any kind of graduate students. So if you have friends or you're looking in the future, definitely have opportunities to come to OSU and work on these problems. But these two and him and her are my newest graduate students on the team, and the others are all undergraduates right now. So I've shared a lot of different stuff with you, but I wanted you to see kind of the scope of different types of things my lab has worked on. And it was a team effort, so it wasn't just me hacking away at lots of different problems, but students who had interest in different things. And some of it was driven by regional or local collaborations. Some of it was driven by opportunities when I met with others that had problems that were approachable to methods I was either interested in or had already developed, or things that were more driven by my own personal interests and application areas. So with that, I'd be happy to take any more questions that you might have about what I've presented today. So thank you.
Thank you. Thanks so much.
Yeah.
Sorry, you can finish.
For this nice overview of everything, including the education. It was very, very nice for you to include that.
So in diabetic kidney disease, do they know why? Like, what is it about the kidney that fails?
So there's about 10,000 of these filtration units, the glomeruli, and some fail. And they know that there's damage to several different cell populations in these filters.
Is
and
it sugar toxicity?
That's a major part of it. And so sugar toxicity, dysregulating them, that then causes them to grow, and then some of that damage is not reversible. So even if you had if you have very tight blood sugar control over long periods of time, you're very unlikely to get this damage. But we're not sure if it's chronic high or if it's peaks of high or if it's too much damage at one time that then is not reversible. Like which are the perturbing dynamics, that's unclear. And so we're trying to probe some of those questions if we have a tool that then is consistent with what data is there. Yes, Cody.
I have a question about the bees. So in the agent-based model, you have them interact with each other, right?
Mm-hmm.
I was wondering if you take away the interaction term, if it modifies like the kind of the global area they were able to go to, or if you guys had investigated that.
The only thing we changed related to their, we had an exclusion volume that was basically, we modeled the centers of each bee, but then we said, okay, a bee body length, they can't physically overlap, though in our simulation or in the videos they can actually overlap because they're physically on vertically on top of each other. But we said it's a 2D environment, so there's no way they could go over each other. But we had the bee body length as a constraint between them. We did turn off this or some of the probabilities of the bumping, like whether that was the same in all cohorts, or if it depended on location, or if it depended on dosing, that kind of thing. And so we saw that we did need to have, or it was statistically significant to have those different groups if they were off the nest or on the nest, and those probabilities of their interaction making a difference in how likely they would be to take off or change their motion style. We did try, we played with that strength of attraction too. That it's actually just a little bit of attractant. It's not a very strong attraction. We thought, well, maybe they mostly ignore each other, but then they might have an influence to the center of the nest or the center of where their neighbors are. If everybody's off the nest, then maybe they want to socially go to where their buddies are. So there's weights for the attraction to their neighbors, attraction to, or the cluster of all the other bees within some cutoff radius, and to the nest, and to a random walk. And so we matched that with experiments, and then once we had that, we left it the same, unless we were trying to tease turning it off or turning it on.
It's like there was a communication effect on like the movements around the nest.
Yeah, and so in the Frontiers paper, my colleague more carefully teased out sort of what he felt was an ecological or more biologically bee relevant reason why they might care about, he called it social buffering, that some of the effects of the pesticide, if there were a lot of bees there, you couldn't be too sedentary because all your neighbors would be bumping against you. And so he argued that some of the proximity of other neighbors would mitigate some of these pesticide effects, and so that there might be different effects you'd see if the nest had more or fewer work, kind of the density of the group. And so he had more of those biological reasonings for why some of the observations we saw with our predictions, but also that were consistent with his experiment. So.
Along those lines, like there is a school of thought that the pesticides affect how bees are interacting with like the sun. So like the photoperiod, it's also affects their directionality, and that also goes into how they interact socially. So would it be possible to include some sort of like you were saying, like a weight, but it's more like an external component to it? I know you said more to keep it local, but
Yeah, so that's a really good point. So our in our model, we just considered this 1 hour time frame that the data was always collected at the same time, and so we don't have time as a driving force. However, in the data that's in the science paper, he did measure that at different times of day and night, and has some clear data about the nighttime regulation, and then also has a piece about the thermal regulation, and they put them outdoors in Massachusetts, so there's various temperature changes that happen, and monitored how well they could thermally regulate. So they had temperature probes, and so they also saw that they were worse at keeping themselves warm when they were under the influence of this pesticide, that they weren't providing the same kind of caps and warming sort of like covers on their nest to the same extent when they were exposed or had been exposed to pesticides. So that's a really good point, and we didn't try to incorporate that computationally, but it's definitely things that there are data for. It could be someone else's problem to work on. Yes?
Can you speak to, very few people try and do this coupling of agent-based models with PD and OD models, and it's a hard thing to do because of their handguns. Can you speak to some of the difficulties that you guys, maybe things that you have learned along the way in trying to couple them?
Yeah, so the software package that we use, CompuCell 3D, it does have a lot of limitations, but we chose it because we wanted to have sort of this hybrid of the approaches, because we knew that there were going to be a lot of chemical fields that wouldn't be that reasonable to do them discretely in the concentrations that they were at, but we wanted some of the advantages of an agent-based approach. So we liked that it did have a coupling between them. It doesn't have very sophisticated PDE solvers though, and it doesn't have any fluid dynamics solvers. It has forward Euler, which those of you familiar with that area would recognize that as the most simple PDE expression, just a change in this over a change in that, and it's very simplistic. And so it has a lot of stability issues, which means we have to take many more time steps. And then we're trying to work on importing different fluids packages that maybe are open source Python fluid solvers that are better and more robust, so that we can have more state-of-the-art solver techniques while coupling them to this agent-based approach. So we're trying to, for our particular problems, think about what algorithms do we need to enhance this other tool that has nice visualization features, has some of the nice integration, but it has weaknesses. It was derived for a different, it was derived for tissues, but not tissues that also had flow in them. And so, so my students are thinking about, what tool do I need to enhance this existing tool to actually solve the problems that we want to. And so we try not to reinvent the wheel, but then if there's a tool we need, we think about how can we make that in a way that's both useful to us, but then we can share with others, because if we have the issue, maybe somebody else does too, or somebody else will in the future. So, so there's those kind of challenges that we're working on, and how do you get those connected in a way that doesn't take forever to solve, and some of the interfaces. So we haven't figured that all out yet, but that's one of our current biggest challenges.
So CompuCell, just to be clear, CompuCell is maintained by Glacier
Mm-hmm.
in Bloomington.
Yes.
Is it called commercial?
It's all open source,
Open source.
as far as I know of. So there's a C platform underneath, but Python and XML on top of that, with these visualization.
He had like a decade of funding to basically have postdocs and semi-professional software engineers building it up. So I think it's been exclusively grant funded in development.
Is it the last one?
Yeah, and they have user trainings for it and stuff, and my students have found them somewhat helpful. But and they're very active live user community. So if you post a question on there, one of the staff will answer a question that day and will point you to pieces and help you debug things. But we just have some needs that we want tools that they don't have, so we're trying to develop those tools and kind of couple it together. So yeah, so.
Yeah, so to make your kidney problem more complicated, and the modeling more complicated, I was thinking that the role of mechanics, with the basement membrane thickening and cross-linking of extracellular matrix might be relevant to what you're looking at.
Yeah. So
Would
we
you have to model that separately? Is it, how would you go about looking at that if you even think that's something worth looking at?
Yeah, so we want the glucose to get the mesangial cells to produce more MMP, to build up more to produce more collagen rather, and that's what will cause these forces on the neighboring cells. And so we can't use ComSol for that, which is what we've been doing for fluid so far, because it won't change the geometry once you've established it. That's why something like CompuCell 3D is attractive, because it would let you have a time-dependent volume constraint or something that depended on another parameter in the system. And so that's where we'd like to go, but we're in year 0 of the project, but that's one of the things we're working on. Definitely. We might end up where that part might be on a different time scale, and so it might be kind of decoupled in time simulations, but that could represent sort of, here's the slow processes that are involved, and here are the fast processes that are involved. But we'd like for it all to work together. Yes.
The kidney's super complicated because it's got the flow and mechanics that's completely complicated. What aspect of kidney dynamics do you think is most important in order to understand or study this problem of diabetes-induced failure?
Yeah,
Because
so
like flow, you mentioned like hand-wringing over trying to get flow properly dealt with. Do you think that flow is an important aspect in the course of basically failure, for example?
So we need just to get the glucose there in our part, but the cells get damaged by the concentration of glucose, and then they play a role in mediating the blood flow that then can lead to no flow through certain sections and more flow through other sections or in different stages of the disease. But the onset is cellular damage, and it's upstream cellular damage in these glomeruli. Some people look at this reabsorption in the tubule that's downstream of the glomerulus, and yes, that damage happens, but it only happens once the damage happens to the glomerulus. So we've focused on the glomerulus as a key factor for the onset, and if it If you prevent more damage to that, you prevent more of everything else. And so that's where we're starting. The flow part, there are other models about ways that flow is regulated. And so one of my former collaborators, her lab mostly focuses on blood flow in the kidney. And so she's actively working on sort of whole kidney types of challenges related to flow. And then we're working, yeah. And so she and I collaborated on the first two publications I put for, or actually the bottom two, but on the physiology part for blood flow regulation. But she doesn't look a lot at diabetes unless it's tubular reabsorption part. So I'm very focused on this combination of biomechanics and biochemistry in the glomerulus in particular. So, or the glomeruli, all of the glomeruli. So we have intentions of, idea of future of seeing what happens if you have populations of them and some of them are failing and some of them are not, and that changing the blood flow and how did that all work together in a larger system too. So we just start very challenging. Yes.
Yeah, I think you can kind of expand a lot of this towards maybe K-12 crowds and stuff like that. I know that you talked about using those GUIs with kids over at the women's outreach thing. But yeah, I guess how would you suggest maybe going maybe a step further with the outreach for this, or maybe bringing it to the classrooms?
We also have taken the idea and instead of using computers but still explaining some of the processes, I have a student who built some cylinders that have holes in them, and we have some beads of different sizes that represent different types of things in the bloodstream. And so if those holes are different sizes, then different things get through and escape and could be part of the urine. And so we talk about how the disease can change the shape and size of the holes in these filters in our kidney that would influence how much can get out. So they can physically shake these tubes that have holes in them of different sizes and can see the beads that fall out, and then we can tell them what the beads represent. So we have some of that stuff. We also do some experiments with water filtration, but we use that as a description for what's happening in the kidney. So we have things with cotton balls as a filter, and that's we mix together food coloring and cornmeal, and we filter that through, and they can see that the cotton ball does a solid separation, but it doesn't remove the coloring. And then we can put it through activated carbon, and we can see that there's a chemical interaction there that's removing the color from that colored water, and so that the kidney has both physical separation processes, but also has a chemical charge, or so not just size exclusion, but some chemical exclusions as well. And so we introduce filtration to them, but with that experiment we can both talk about sort of environmental and water quality type things, but we can also talk about biomedical kidney filtration. And then we talk about how in biomedical engineering we may be thinking about both models of these, but how do we engineer different devices that could be an internal replacement kidney, an external replacement kidney, or something like dialysis machines. What are some trade-offs with that? And especially with the grandparent university, some of the kids have grandparents on dialysis who may or may not be at the camp, but it kind of gets the age empathy thing where they can talk about issues that plague their family when someone has to undergo dialysis and how that actually affects them as a kid if grandma can't go to all the events when they're on vacation because they have to go to a dialysis center during part of the time. And, so that's ways that we've also used some of these ideas, but maybe not a computer simulation.
So also a quick question about the educational part. So the PDK model that you present for hypertension.
Mm-hmm.
And so you presented like an educational tool, like a dosage, you know, the how have you sort of compared what you were doing with what the hospital industry does? In other words, they also use like a chemical engineering approach, you know, when they think about PDPK, because for my little interaction with pharmacologists, they don't seem to be thinking that way.
I think the ones that do quantitative systems pharmacology do pretty similar types of approaches. There's some classic pharmacology textbooks, and their chapter on PK fits very nicely with the chemical engineering, chemical kinetics type of book. They just work out all the analytical solutions, versus the chemi book would keep it as ODEs so that you could then solve them simultaneously. And the pharma book would have, here's the solution, here's what the shape looks like. But there's a number of software packages that allow them to automate some of those processes, so they don't have to be well versed in differential equations, but they can put in certain kinetic measurements, or like, here's the shape of this curve, I think it's first order, and they can extract certain things. And so again, not necessarily teaching all the math, but can they do something of kind of classic types of profiles and have automated computer tools that pull out some of the pieces for them? Those are actually quite common. And so one of my students who just graduated, she's actually working in a pharmaceutical company now, but she's building models for disease states and how different populations interact, or different people in different sections of the population, or how different, if you have variations in different types of, like you might have a different response in some way, how does that stochasticity maybe influence the changes in drugs? And so I think there are some that are of the same philosophy as me and us, but they are being infused into the pharmaceutical industry now.
Terminology, quantitative systems pharmacology.
Mhm. It's definitely a big buzzword these days, and there are whole papers about that, or whole journals about systems pharmacology. So my student actually applied for positions called Systems Pharmacology or Center of Quantitative Systems Pharmacology. And so this type of degree program that some of you are in could be a really good fit for those types of industrial jobs. And so they have experimental and computational people working together with quantitative systems, but applying them to pharmaceutical systems. So.
It's a buzzword that's being fleshed out.
Mm-hmm. Yeah.
All right, anything else? Okay, so thank you, Ashley.
Thank you.